pesquisador do Instituto Federal de Pesquisa Aquáticas, que é da Suíça, que é vinculado ao Ministério de Ciência e Tecnologia na Suíça, e tem trabalhado já há muitos anos com a modelagem computacional e matemática de sistemas ecológicos e evolutivos. E nos falará hoje sobre Eco-devo-evo, dinâmica da convergência, divergência e metacomunidades. Queria muito agradecer ao Carlos por estar conosco, e temos uma questão para a audiência, se vocês preferiram que o Carlos fale em espanhol ou em inglês.
I don't speak Portuguese, I'm sorry. I would like, but...
Então, inglês ou espanhol?
English.
Inglês, quem levanta a mão aí? Inglês. Espanhol. Olha o empate.
Half in Spanish and half in English.
You choose.
Flip a coin.
Es difícil, ¿no? No sé. Yo hablo despacio español. Quizás English maybe is a bit faster. No sé en qué idioma me... O sea, most of the concepts I'm going to discuss today, I'm more familiar in English, I have to tell you. Maybe, yeah, it's better maybe. And the reason is I grew up in English, scientifically speaking, and maybe, I mean, some of you are maybe feeling the same. So I'm going to try to be quick. Stop me anytime. It's quite, you know, this is quite, I think, informal talk. Indeed, it's an unexpected talk. I didn't expect to give a talk today, till a few days ago, so it's quite improvised. And it's a quite difficult topic. I mean, most of you or most, I mean, feel that this title is quite, you know, surrealistic at some point. And most of these results are still, this is work in progress. So I still don't know the robustness of these results. We are still doing more simulations, and I think this is important to know, right? Because it's part of this, you know, improvisation. So as you can see on the top, just 4 parts. It's a quite short talk, so maybe, yeah, let's see if we move forward quickly. I'm gonna start with an animation. I have to tell you, if I were able to do an animation for any slide here, I would prefer to do so. Just sit down and just animations, and then we discuss, and that's it. This is my way to do science. Is doing modeling, animations, we learn through visualization and equations and a lot of discussion. And it's the way we move forward, a lot of new questions. So most of the time my words are gonna be a bit out of the blue. And the main points are the animations, and then you feel more or less why, for example, in this talk today, why Eco-devo-evo? Why? So this is just a quick illustration. You can imagine anything from here, from art, but also individuals in a space. Each dot is an individual here, for example, and then each color is the background. This is the idea of propensity. Indeed, we were discussing before quite deep discussions, philosophical terms, but also scientific terms. And here, propensity is that what one individual does is essentially a function of the context. In this case, it's a color, the background. It's like a habitat, right? Driven by temperature or, you know, acidity, anything you can have in mind, also several variables. But the main point here is that the individuals, they are connected, so they maybe know each other, they can mate, they can have vial offspring, but then the landscape is also fluctuating, right? These connections are fluctuating. It's a cartoon. It's quite simple stuff, but this is mostly happening, right, at almost any level and scale. You have interacting things, right? And this is the whole idea of this Eco-devo-evo, right? Is how to put interacting things at different levels and spatial scales in the same framework. We can do this by art, but also with our computers, with equations. We can do this from a lot of different things. So I'm sure you see new things here, but this whole kind of problems have been long discussed in science. For example, first thing, wrote down a quite classic paper. I recommend most of you to read this paper. It's beautiful because it illustrates classical problems, right, in speciation theory. So this is more or less introduction. There is a seat here. Okay, wanna sit down? Okay. I don't know where I put my eyes. Is there? So should we move backwards or forward? Because most people now is coming in, maybe forward, right? So we were asking why Eco-devo-evo, and this is one of the classical model systems in adaptive radiations. So these are the secrets from Eastern Africa. So these guys, you know, in some lakes you have like in the last maybe 1 million years, even less, 200 species from 1 or 2 founder effects. 1, 2 founder populations arrived less than 1 million ago. Today 200 species. That's huge. For some lakes, and in other lakes, even in the same latitude, no speciation at all. It's quite amazing the geographic variation of these guys. But the main point I want to illustrate here is that when I first was learning about these guys, I thought with this picture, okay, that's quite clear, right? These guys are related phylogenetically in rows, right? More than in the columns. But this is not true. And so then I said, wow. So these guys are more related phylogenetically than these guys in horizontal. And so the point is, why? So is this adaptations to niches? Is this adaptation to the different conditions, right? The lake. Also additional genetic mechanisms. But is this because development? So this is still open, and there are a lot of things open in this, right? Exploring these guys. But the main point here is that maybe we need to take into account at least some of the genetics and development and the ecology of these guys to understand what are the drivers, right, that produce such a phenotypic variation, right, within the same lake. And so this is something we have been working, but today I'm going to show more or less some modeling approach, but we are not still, we are far away from answering, right, or understanding the mechanisms driving these right, explosions of diversity. But today is like a toy model to discuss with you. Maybe you can also criticize a bit some of the assumptions I'm going to discuss with you. But the main goal in the long term, I don't know, 5, 10 years, the idea is to really have predictions, testable predictions, a set of scenarios where we can really compare the outputs from the models with the data at genetic and development and also at ecological levels simultaneously. But it's a long-term goal. So I'm going to show you now more or less how we are trying to approach this problem. We can, for example, this is classical approximation for people that do population genetics, is that you have, this is quite a cartoon also. You can visualize almost anything, but the main point here is you have dots. These are individuals, and they are expanding from this corner to another corner. It's like a population that is expanding because some, you know, processes, maybe they are tracking fluctuating environment. But the main point also here is that there are several processes. Here we have birth and death. We pick up the parents from the local neighborhood, right? We recombine and we have mutation rates and we have an offspring and we have this individual that is moving with some reference in some direction. And at the same time, each time we have a mutation, we put a color, right? Randomly. No selection here. Not in principle, it's just neutral. This is a neutral landscape. Individuals move in space, and as you can see, they reach the other corner. And most of these individuals, they have a different phenotype. So this is more quantitative genetics approach, right? So the changes at genetic level change, right? Some phenotypic aspects. This is quite classical approach. So here, we can also, for example, visualize this. Yeah, these fluctuations is the classical problem between Linux and Windows, but we can visualize this in this way. So it's like a time, and this is the frequency of the phenotypes. And so you can, for example, you have to define a phenotype. This is an operative definition. A color or some portion of the genome, right? You define phenotypes and you track them in time, and they fluctuate, and most of them, as you can see indeed, they have pretty low abundance and they go to extinction. So this is within one population that is diverging in time. You have several also phenotype formation, and some of them go to extinction and others they fixed in the population. This is nothing new for people who do population genetics. It's a standard, I think most. But the point is that now we want to extend, we want to go beyond, right? Just these classical approaches. So how should we do this? So this is the next and most, at least to me, challenging question, is how now okay, so given this background, now we can more or less focus, right, a bit the talk and say, okay, do ecological, genetic, and developmental drift predict observed convergence in metacommunities? We don't know nothing. I mean, we know nothing about this. Right? So, because first, no models taking into account these three elements, just in the simplest scenario is drift. So no selection, no habitat heterogeneity. I mean, it's like the backbone of a model of this kind. But the main point I'm going to illustrate here is not only these three elements. It's also that until now I have been discussing in the context of one population. But now I just come to meta-communities. And this implies several species in space, not just one. And the point is how we scale from a population that diverges in space to several species that normally we see of the same kind of type of species in spatial landscapes. For most communities, we see several species. So this is a big problem, right? And so this is essentially the problem of speciation, and it's a quite difficult problem. To me, is the most difficult concept to model. It's quite challenging to model speciation. And I'm not saying only here speciation at the species level, but at the molecular level, or at any level we have formation of new things. And to tracking these things, given a set of processes, to me is quite challenging. So the main point here then is how we go from these processes within populations and how we scale up to meta-communities. And the second point I'm going to kind of illustrate today also is that this can be helpful, these kind of models can help us a bit to also scale up to macroevolution and also biogeography. So we put individuals in space, a set of rules, and we start to see divergence, and also at some point, given some definition, operative definition of speciation, we start to see a species, and then at some point you have several species, and this is the idea of biogeography, right? Species expanding and contracting space and time. And after a long time, we can track where most speciation events and extinction events have been happening through all these, right, time. And this is essentially the idea of hot and cold spots. And this is also, I think, also interesting in not only in ecology and evolution, but also in innovation or even technology of societies. How or what are the mechanisms we have to put together to generate hotspots of new ideas? This is a hot topic today. because it's quite challenging, right? Most people, most society, they want to bring a lot of things together and develop big thinking and new innovation, a lot of things, right? So how can we go or scale up from these models to anticipate the hot and cold spot formation? I'm going to try to also give some preliminary results about these problems. So I'm going to describe briefly the model we have been working with, and then so I'm going to go more or less with some detail through it, and then I'm going to present some results from this model and move forward to extensions of this model to understand better hot and cold spots. And so here we are going to, this is a cartoon where essentially we have all these from A to F steps, and this is happening at any given time step. It's like we go to a maximum level of detail, right? And we try to understand what we are doing with the code. So what we are essentially having is we put individuals in a space. We can put individuals in a space following different rules. In the next set of slides, I'm going to show you also how, right? Maybe some of you even know, I'm sure more than me, how to generate different kind of landscapes. And so, but for this cartoon, you put just a few individuals, right? They have different colors, maybe different phenotypes or genotypes. But the point I wanted to illustrate here is that you normally kill randomly one individual. This is the empty spot. Now here is a cartoon where we are coupling birth and death, a demographic step. So you kill one individual randomly, and then you pick up, this is step A. In step B, you have sexual reproduction, right, between one male and female, if they satisfy some conditions. We have put several rules here, but just think it's like we are going an incredible level of detail. And this is, I would like to stop quickly here, because this is something, these are individual-based models. Most people working in general in theoretical ecology, this is quite a level of description where normally most people is not thinking every day at this level. It's thinking more in means populations, infinite population sizes, and Lotka-Volterra equations. The idea here is why individual-based models, we put individuals in a space, individuals contain genomes. I'm going to discuss more or less how we deal with the genomes, and you can put also some phenotypes to these individuals, a set of traits. This is already a lot, even if it's a cartoon, and it's maybe quite simplified for some of you that work with the specific parts of these different steps. Maybe you say, okay, you are simplifying a lot. Yes, but the problem is how to put several simplified process in a coherent framework. And the point here is you have then sexual reproduction if you satisfy these two conditions. The first condition is these two guys are within a distance, a geographic distance. This is a tunable parameter. Is D max. And the second condition, if they are sufficiently similar, then they can generate viable offspring. So this means that this is a threshold, and in this model, assumption is that this incompatibility threshold is endogenous. It's something that because some individuals have been diverging in space, at some point, if they, because different reasons, they are again within this maximal distance, they can mate, but they are not similarly enough to generate viable offspring. They can mate, but the offspring is not viable. This is the assumption of the model. There are a lot of potential assumptions, but the whole idea here is that then, as soon as you have sexual reproduction, you have the two genomes for this individual, and then you recombine them is the first step. The crossover type of recombination is the simplest, and then in the last step we have we go through the whole genome of the offspring, and we have mutations with some probability. This is C and D, and in the last step, this individual I, the offspring, then is essentially has a genome from the two parents plus a phenotype. This is the idea of development in this model. It's quite simple. But the idea is that this guy contains a phenotype that is essentially inherited from his parents and plus some phenotypic plasticity. And then we go backwards and we fill the spot here. This is one cycle. One generation, normally, this is an overlapping generation model, is when you do this for all the individuals in the population. And so we move forward the model and we can try to derive several predictions. I would like to emphasize here a few things. The first thing I would like to emphasize is this is the simplest set of processes, I would say. And it is not capturing most of the biology today, right, known for several of these elements. And despite that, it's terrible. It's terrible. I mean, terrible in the sense it's really difficult to model these things, for example, for 1 million individuals.
You're assuming that they are diploid?
Yes, we are assuming haploid individuals. With diploid, it's easy, right? But then computation time, increases for each generation, for example, because you have to do an additional steps at this level of detail. So individuals, instead, for example, of 1 million, maybe 500,000, and if you add more complexity or more realism, we are paying a cost to produce more and more replicates and to be able to have predictions and compare the predictions with the data. This, I would like to say, another point here is, at least to me, is the boundary between science and philosophy, and also science and different fields of science. If I specialize as a scientist in a specific field, I would say this is bullshit. Because what I'm doing is going deeper and I understand more of one specific things within these processes. This is a real problem in science today. I'm not saying it's bad or good, but it's a problem because people that is trying to do hybrid science or integrative science is facing a lot of penalizations. This is a hardcore problem. Maybe we can discuss this more in detail, but this is a hardcore problem in science. So when you try to put different things together, you must, or unless you do something really novel and you do how to combine different things in deep, if you cannot do that, then you have to simplify all the things, and at least you are putting something together, but you are simplifying a lot. This is a classical trade-off that I think most of us face in our very everyday life. This is not new for you, but this is also happening here. Just to move forward, what we now do is we run generations, millions, thousands of generations, given this set of processes. So we put individuals in a space. Each dot is an individual. It's a fully connected graph. It's like this previous animation. Individuals, they invade a lake. The foundry effect. Let's say these individuals are all exactly the same. They are relatives, no standing genetic variation, and they arrive to a lake. And now we want to start to do the simulations, and we start to have on the periphery of the network diverging populations, just by drift. So this is a neutral landscape. So we can also say, well, but this is because these guys, they invaded the lake, but also with depth, they started to acquire, new mutations, and then they start to have higher fitness in those situations. But this is not the case for this model. Indeed, the only way to detect the strength of niche-driven divergence and speciation is to compare this data, the empirical data, with the predictions from a neutral model. Then is where you can really distinguish, right? You can extract, infer the strength of selection, right, required to have the observed pattern of divergence. So this is one of the reasons of these neutral models. And so the point is that you move forward a simulation. It's a cartoon to add the concept that we still need to understand how we scale up from populations to metacommunities. And it's essentially when you move forward, you have to define a speciation. And we define a speciation here at some point is an operative definition, because there are potentially several definitions of speciation, but what we are doing here is, as I mentioned before, we have as a minimum genetic similarity value, that is a threshold, and it's essentially telling the conditions to have viable offspring. And using this approach, this is the genetic similarity matrix, and you can essentially, given this genetic similarity value, that is a parameter, you can distinguish when you have different components in this population. This is essentially the idea of a species. These three individuals can make, you have indeed individuals within the same species. For example, let's say in this population, these two individuals, they belong to the same species, but they cannot mate. This is happening in this model. So some individuals belong to the same population, but they cannot produce fertile offspring. And so this is essentially the idea of speciation. You move forward the simulations, and you are going to have components or new species, and they are going to be drifting, fluctuating, and some of them go to extinction quickly, and others they become common, and at some point even become dominant in some specific conditions. This is the idea of genetic drift scaling with ecological drift. So you have alleles fluctuating within populations, but you also have populations fluctuating within communities. But in addition to that, you have populations fluctuating within meta-communities, because at some point you can have, for the same species, you can have species that fluctuate in different parts of the space. We can track these things with this kind of approach. So even if this is a really simple model, it looks quite complicated from the coding point of view. It's quite challenging and demanding, which is frustrating sometimes. So I'm going to show you maybe one concept that we can quantify from this model. Is the idea of evolutionary convergence. So it's quite simple. So we have, for example, two matrices. We can track the genetic similarity matrix and values, and the phenotypic similarity matrix for three species, A, B, C. And at some point, after several replicates, we can more or less say the similarity between each pair of species. And more or less, you can then start to analyze how genetic similarity correlates with phenotypic similarity. And normally you expect a high correlation, but the main point here is that you can control, in some scenarios, the idea of evolutionary convergence is that you can draw a tree and you can then control for genetic similarity. And for example, you can define evolutionary convergence like you discard the species that are sister species. And then you go, for example, here you compare A and B and C and B. And in this scenario, you can have A and B, they have been diverging a lot genetically, but they are quite similar phenotypically. So this is genetic divergence but phenotypic convergence. This can happen in this kind of models, just by drift, no selection at all. And the point is that we can have a prediction to for example, to say, are we able to predict the divergence, phenotypic divergence in meta-communities just by drift? Or the data is telling us that the convergence is beyond the confidence interval that we can draw from the neutral expectation. This is more interesting because the tradition in ecology and evolution is that convergence is mostly driven by habitat, similarity in habitats. So you have populations that arrive to habitats with the same kind of conditions, and then these populations, they diverge at some level, but also they converge because they are adapted to the same kind of situations. But we can have a neutral expectation, that is that this convergence is not driven to adaptations to these similar conditions, but just because neutral processes. This is the whole idea of neutral versus selection. So we can have a prediction and a neutral expectation. So for example here, these are two different replicates. We have initial trait distribution, and after several replicates, we have a more or less steady state finite trait distribution. These are all individuals, right? And each has a specific trait value. This trait can represent several things. The length of the proboscis, for example, for pollinators, but can also be the number of gill rays for fish. Can be important traits that are determining, for example, how they have access to resources. We can also have think in different kind of traits, but the main point here is that because we are tracking species, we can plot, for example, the mean and the variance, right, for this distribution for each species. For example, here the most common contains the largest variance, and then you are more and more rare, right? You are diverging more and the variance is much lower. But as you can see also here, that the trait convergence is huge across species for these two replicates. We can generate hundreds of thousands of replicates, and then, as I mentioned before, we can have a neutral expectation and compare with observed data. For this kind of model, we're still working with this. We're trying to make it faster and to have a sufficient number of replicates to be able to generate the statistics and compare the outputs with data.
How is the relationship between the genotype and the phenotype in your model?
Oh. So here we have only one scenario in this model, so not two scenarios with different options, which is something we are thinking now. But is essentially the phenotype of the new individual, I, is essentially the genotype inherited from his parents, plus a normal distribution with some mean and variance. This is essentially the phenotype.
What you can change is the variability of this distribution.
It's essentially saying that when you are living in a highly fluctuating environment, let's say the variance is huge, and then plasticity, moves to the variance. You can have individuals, highly variable individuals, and the opposite, you have, let's say, highly predictable environments, and then maybe we move down the variance, and the mean is capturing most of. But this is a set of assumptions, right?
I'm asking about the assumptions. The relationship between genotype, the variance of the phenotype is connected to the genotype, or is connected to the environment?
Good point. So we don't have explicit environment fluctuation in the model.
Because it's neutral.
That's one of the reasons.
Plain.
Yes. You can add environmental fluctuations also in neutral landscapes, right? You can have fluctuations, but individuals can maintain the fitness, right? It's like you are in a lake, and in some portion of the column, in the water column, and you are in a state of the lake, state A, and you have some phenotypes, and then you change to state B, and you can have the same phenotypes in the model point of view. In the data, normally, you have new phenotypes arriving to state B or state C. You can define as many states, that essentially each state is an environmental state. But we don't have this in this model. The environmental state is always the same. Yeah.
So it's a property of the genotype?
It's a property of the genotype plus some phenotypic plasticity.
Which you define as a parameter you can change?
Yes.
I was just going to say that's something like developmental norms, like the bird.
You think it's more or less a similar concept?
It seems like developmental norms.
But without, what I see a bit, we are now discussing with people working with development, and here is also something interesting to discuss these days, is how to implement a more realistic, right, development here. For example, using gene regulatory networks, something like that, where you have several paths, and you more or less define a few set of rules, and you can pick up with more or less probability some of these phenotypes according with the state of the gene regulatory network. But maybe this is going a bit right? Complicated, given already the set of processes.
It's almost a neutral model of development.
Yes, it's like developmental drift. Let's say developmental drift. This is the idea. Yes. That's a good. So we are almost done. This is the part where I have been working most lately. And so it's essentially extending the model, but just cancelling the phenotypic part. We cancel to zero the mean and the variance. So no phenotypes here. But the model is essentially the same. And now we can, for example, using random geometric graphs, we can generate landscapes. And the idea here is each red dot is a population. Can contain 10,000 individuals or some number of individuals. And then you just use one parameter, the radius. Use one parameter to define how you connect each two sites, right? If the two sites, two randomly selected sites, the geographic distance is below their critical radius, right? Then you connect them. If not, you don't connect them. That's it. It's quite easy, but the point here is you generate several kinds of landscapes. From the infinite island model, or where you have all the sites are disconnected, to the other extreme where you have almost fully connected graph. This is more or less a cartoon with 1,000 times 1,000 kilometers. But the point here now is, okay, under which conditions can we produce in neutral landscapes a hot and a cold spot? Is this possible? By hot spot here is where most speciation events, no, I'm wrong here, where diversification rate is the highest, or is higher in comparison with other parts of the landscape. Is where you are having several speciation events and some extinction events, but in the balance, the net diversification rate, right, is higher. And so here, because we track speciation events and extinction events, but now in each portion of the spatial landscape, we know where these events are happening. It's quite a painful code because you have to track where the speciation events are happening. And from the coding point of view, you have to tell, well, if we do this each generation, we are going to take a lot. Because each generation, you have to go through the whole matrix and detect the components. But you can also do this, maybe, let's say, each 10 generations, 100 generations, and then check which is the degree of resolution that is telling you, right, the real diversification rate. And so the point here is that now we can run again the same model. Individuals, invade this network, and now they move across the network, and then they start to diverge. And we want to know where the speciation events and the extinction events are happening. And now, here we are doing essentially, we are trying to infer two kinds of mechanisms. One is the landscape structure, and the second is essentially the gene flow, how gene flow move in space. And so we have three models now. One is symmetric gene flow. This is essentially that regardless the landscape, individuals, they move in any direction with the same probability. For example, in here, well, they don't move because we don't have indeed migration, right? You have a birth death, but the demographic stochasticity only happening within sites. No migration. Here, you have some migration, some sites are connected, others they are not, but normally we do the same processes than the process I was kind of describing before, but in this kind of landscapes. And, sorry, the point is that when you have symmetric gene flow, we have been doing several simulations, several replicates, we never observe the formation of hot and cold spots. So speciation rates are quite homogeneous through the landscape. And extinction rates also, so diversification rate is quite homogeneous across the landscape. This is quite expected for several reasons. But then we have two additional models. We wanted to explore, for example, periphery to center gene flow Well, essentially we are saying that individuals are moving from the periphery to the center. So when we pick up individuals and we have to put the offspring in some part of the space, right, we are tracking total distance of each site with respect to all other sites in the network. And we know where those sites are. This is essentially the total distance of each site. And so when we take this into account, we know that in this model, individuals are essentially moving from the periphery to the center. Most people say, but why? Well, this is just an assumption. Maybe there are several reasons for that. Populations are contracting, or the center is more attractive because more resources, or less predators. There are potential several reasons for this to happen. But this is just a phenomenological model. We assume the individuals are moving essentially from the periphery to the center. And here is the opposite. Individuals are moving from the center to the periphery. It's maybe density-dependent effect, and so you need to move to outside to the periphery to the network. And the point we want to illustrate here, and now we combine this model, periphery to center and center to periphery, in these different landscapes. And so what we would expect is essentially more or less what I'm going to explain you briefly here. So we, after hundreds of replicates, we calculate the center of the landscape to the periphery, right? Like slices. We cut slices of the graph, and now we say, okay, we plot the diversification rate, right? Given the three models. This is periphery to center model. You see the highest, this is the mean after all the replicates we run, and the highest diversification rate, and then you go down, down, down to the periphery. And the center to periphery, you also get a hotspot formation in the periphery, but is maybe half, around half the other model. And in the third model with symmetric gene flow, regardless the landscape, you never get a hotspot. There is an additional element here that these two kind of hotspots in the center or in the periphery, they only are produced here in the center. They never produce in the extreme of these networks. We are now trying to understand this. Maybe you know some. The main points seem to be related with the diversity of the gene pool that you are moving around the network. So when you have an isolated set of islands or sites, you never are bringing new genetic pool. So you have speciation events, but they are similar across the whole landscape. I think most of us can visualize this. In the other extreme, you have almost a fully connected graph, and what you are doing essentially is the network has such a level of connectivity that you are essentially moving almost the same gene flow across the whole network. But in the center, you are maximizing the diversity of the gene pool, in the sense that if you pick up randomly a site and it's connected with different components of the network, these components contain each a highly diversified set of gene pool. And all these gene pools coming to the center increases the speed at which new species form, and you have high diversification rates. This is essentially the explanation, but maybe we still are running simulations to understand why hotspots are essentially occurring in this state. This is essentially the percolation, close to the percolation threshold. For people doing physics, this is quite a well-known state. And it's a state where most metacommunities, by a metacommunity I mean some portion of the network, they are almost isolated. There are a few connections with other metacommunities, but most of them can be almost totally isolated. This is the so hotspots occur nearly the percolation threshold in random geometric graphs. So we can visualize this like using this way of visualizing. So you have in the periphery to center model, in the periphery hot spot, in the center you have a hot spot most species on events, diversification rate is the highest in the center. And the center to periphery, you have the diversification rate is highest in the periphery. But as you can see, there is some kind of asymmetry. And one of the reasons is what I said now. The center, you have this diversified pool coming from different places. In the periphery, you have the same effect, but in the opposite direction. You only, in two-dimensional landscape, you only have the pool is not coming from the whole surrounding, but only from a portion of the network. And so you never get as the diversification rate that you get normally in the center. In the symmetric conditions, or where we have states, the two extremes of the networks, you never get really a hot spot. This is the whole idea of this approach. So just to summarize, so this is like an incipient stage of a non-adaptive Eco-devo-evo model to connect landscape genetics, development, and ecological processes. So just still working on this kind of result, tried convergence can be reproduced by ecological, developmental, and genetic drift in absence of selection. We still don't know whether the predictions from this model are able to really capture the empirical data. This is, I think, the next step that we should do. Concerning the hot spots, so landscape structure and directionality of inflow, both combined, may play a critical role to anticipate the formation of hot and cold spots. And so yet, we are far to have robust predictions with realistic landscapes to compare with the empirical observations. And that's it. Thank you.
I enjoyed your show. At the same time, it's interesting that you present your model as based on drift, since for me the critical feature is associative mating.
Of the model?
Yes.
As a
it's associative mating,
yes,
which triggers the divergence.
Indeed.
And what we could expect from a pure neutral model to see the patterns which emerge without associative.
How would you do that?
It's interesting that you...
And how would you do that? This is sorry. How do you think should we take into account neutral without a sort of limit? This is a good point. So in Palmitic populations,
yes, you give up the assumptions of because it's interesting that the only paper you quoted was Felsenstein's paper, and his objection to symbiotic speciation, if I remember, in this model,
yes,
was this associative mating. You have one type which chooses the opposite type,
yes,
and this prevents symbiotic speciation.
Yes.
So I think in the presentation of the model and its implication, it's good to emphasize that the critical
point, right?
Point is
yes,
associative mating, and because it matters, Felsenstein's point was not totally absurd, because in Drosophila we know that mate choice is always for the rare male. When you are a female, you can choose to choose the rare male. Here in your model, a rare male is doomed to extinction in a phenotype.
So you mean, yeah, this is quite an interesting thing. So I see 2 points here. One, I didn't emphasize assortative mating essentially because, as Sergei Gavilet said, for example, we, within a given location, we are not really taking into account phenotypes. You can make within a given location with any individual that satisfies the conditions, regardless of the phenotype. You see, but your point is interesting. We could also add, given a location, you have the information of all individuals, red guys, and now you do like a frequency-dependent mating scheme, and you have the frequency of each phenotype, and you track those. So in addition to local mating, you have explicit assortative mating. Am I correct in this?
Yes, but this would be another model testing something else.
Yes.
So it's not related to your point.
Yes, but we have been thinking of this, and it's quite interesting, right? For example, adding explicitly, as in the first model I described, you have the phenotypes there, right? And then you can track the phenotypes, and you can emphasize, when mating, you can emphasize the phenotypes that have some advantages, like the Drosophila example. We have been thinking this, and how this changed the pattern, for example, of speciation, right? I mean, this is, yeah. But I agree that maybe I should emphasize a bit more about the assortative mating aspect of the model. Yes, yeah.
Mariano.
Yes.
Thank you for your speech. And I'd like to ask you, the way I saw, in fact, one kind of structure that you change in the spatial structure of the population was there are 3 types of population. Extremely integrated,
Yes.
more or less integrated, and the ones that were isolated.
Yes.
And in fact, the question is how far we are from introduce here a kind of real landscape spatial structures. You know, we have a central population. You know, if we have a landscape with a large patch of habitat surrounded by small patches of habitat, how far we are to put your model in a real.
And you will respond to this question. Maybe Charles.
Tomorrow.
This is a great question. We are working, but it's still not. But we are trying to say, given a realistic landscape, for example, today, continental drift data is available. Maybe 1 million year resolution, something like that. I don't remember. 1 million. So you can add explicitly. Maybe this is explicitly the idea. You can have these models in, in some portion of on Earth, and then you have drift, continental drift. So yeah, additional drift. You have these 3 kind of drifts plus continental drift. And so you now run these simulations and you start to count the speciation events.
You're asking because we have in some students, some work that we are doing now, that we are now gathering data on genetic diversity, on genetic signature of individuals
Yes.
in landscapes, and we are measuring also the genetic flux by the pattern fatherhood tests.
Yes. But you have already populations in
Yes, we have
space.
population, and we have this in several types of landscapes. And we infer that some processes are interfering in genetic flow.
Which kind of data do you have for the landscapes? You have the topography, for example?
Yes, and the distribution of the habitat types that we think that they are related to the movement of the dispersal.
Because habitat preferences, for example.
Yes. And in fact, we are measuring the genetic diversity of the saplings of some trees that are dispersed by bats, and in the kind of environment that we have several types of landscapes that they have different arrangements, different arrange of patches.
And how big is the landscape? How many kilometers is
The entire landscape,
Yes.
or the area?
the sampling area, more or less?
Sampling area, more or less 1 to 2 square kilometers.
Okay.
We choose this because of the range of the bats flying. And the entire region that we are working with, and we have this landscape measure, is about 50 square kilometers.
Okay.
So something else to do in Pronex in collaboration with Project 2. So, Hilton.
I was just wondering, you have a pretty neutral evolutionary model connected with kind of a neutral ecological model, because the interactions between the species, they don't matter.
It's symmetric.
Yes.
It's symmetric.
So it's, but I was wondering, what is a neutral developmental model? Because what would be a development, what would be the, for example, selection is a key important process in evolution, while interactions are key important factors in ecological communities. And development, what are the key that I could take out and see what happens in the absence of this key factor? And maybe the interactions between the genes, for example, stasia, or these, or the interactions with the environment are the key factors. And so I'm just trying to see again if I agree that you have a neutral developmental model or not. And so I think we have to think about this.
That's a good, yeah. I mean, two minor things here. I agree. I think the developmental model or the developmental part of this model should be improved, right? This is the first point. And the second point is selection, let's say, is the main thing. But the strength of selection can only be obtained when you compare with, we agree in that also. If not, we assume as scientists that we cannot say, yes, selection is everything, but you have to test, right, how strong it is. This is the point, right? And I see your point here with how to...
What would be a developmental neutral model?
Yes.
Leticia, I'm subscribing myself.
Yes.
So going back to the associative rating, so they need to find a type that is similar enough in order to make it.
A genetic type, not a phenotypic type. This is maybe,
So, but if they don't find a type that is similar enough, then they don't make it.
Good point.
So then it's not neutral, there is negative frequency dependence. The rare types will be selected against because they are not likely to find mates.
So if you track fitness, you track the probability to die and the probability to have a viable offspring, is quite homogeneous in this model. But it's not that all individuals have exactly the same fitness. This is not the point of neutral predictions. So you have variation, of course, but the variation is highly homogeneous. You see the point.
Yeah,
so
but the thing mainly run the types that happen to be rare by chance disappear because they don't find mates.
Yes, indeed, this can happen. This is the idea. You are essentially describing the idea of neutrally, right? Neutral, nearly neutral dynamics. So indeed, some individuals, this graph is going to tell you how many individuals you can make, right? The degree, the number of interactions. And some individuals maybe can make with one, and others with the whole network. This is your point.
Or just that it only depends on the number of individuals of any one time that happen to be there, and those that happen to be rare will not find a mate, and so that type will disappear. So there will be like a species turnover and extinction of the species that are rare.
That's a good point. This is happening there, and it's also happening some kind nearly neutral frequency dependence, but it's not strong. We have been testing. Indeed, we have an alternative model with explicit negative frequency dependence selection. That is essentially rare types that are the types that have less number of connections, increase fitness. This is essentially what we are saying is that these guys, in the productive proteins, they are a highly diversified set of proteins and are less attacked than the common types within each graph. And so then you have real negative frequency dependent selection. Yeah.
Would there be a tendency for similar types to be spatially more closer in space, or they are completely random in there?
Say it again.
Like, would the ones that are similar to each other develop a tendency somehow to be closer to each other in space, or that's not possible?
You mean the genetic similarity?
Yeah, genetic.
If they aggregate in space.
Do they aggregate in space the genetically similar ones, or?
It's a good question. We are trying to map. This is going to depend on gene flow model also, right? Where you speciate. This is the classical. This is an interesting point. Is how diversification rate and biodiversity correspond in space. Right? This is essentially, so maybe you have a lot of speciation events, right, and low extinction rates. Maybe you're going to expect also a lot of species they correspond, but then at some point, because it's a long-term process, you are having migration, right, or moving in some direction, and at some point, diversification and richness decouple in space, and we are trying to understand this. For this model, I cannot tell you, for the hotspot, we are essentially calculating the mean. When you have an speciation event, we calculate the geographic, right, values of each individual, and we do the mean. And the mean is the speciation event. But the mean contains a lot of variance. You see the point? So sometimes you have a speciation event that is around, let's say, 100 sites, and we are doing the mean. It's a good point. It's a good point.
And then the number of species is super proportional to the population size.
The number of speciation events.
Species that emerge.
Yes.
Is that quite related to the total population size?
Yes. In these models, speciation rate is correlated with population size. That's a good point.
And that might explain the hotspot, because also the populations get bigger at the center if there's migration towards the center.
That's a good point, but here what I see is that when you have a hotspot in the center, migrants are coming, let's say, from the most dominant species, but from different parts of the landscape, right? So the gene pool is highly diversified. Indeed, the same species after several generations, maybe it's a different species. But the pool, they were the same species, they have moving around, and at some point, they are now going to the center, but they form a different genetic pool. They belong to a new species, or they are highly diversified at genetic level. But these are great questions. I'm running simulations with these codes these days, and I still don't understand a lot of things. Yeah.
Let me ask you, you didn't implement genetic regulatory networks to implement developmental drift.
No.
So the only thing you are doing is that if development would be a sphere, the ball can move anywhere here with any probability. So they can change in development for any direction, any tendency, no preferred.
When we have a high variance, yes, we have the genotype, and then a high variance, you change the quantitative, the phenotypic trait, right? And then the variance within a given population is going to expand. Because in some time you have an individual with a low trait value, another time you have a really high value, and they all belong to the same species. Because you see them, but you are expanding the phenotypic variance. Our argument is essentially related with fluctuating environments. So genetic plasticity, or what you mentioned before, right? The developmental, yes, is expanding in that situation, but it's phenomenological. So we are not really saying, given that these guys contain a gene regulatory network that is expressing with more probability.
But my question is, why would you need that? Because Newton was asking what you should cancel to have a system which is developmentally drifting, and maybe you just can do it phenomenologically. I don't see why you have to complexify the model, introduce a GRN if you are getting something that is drifting developmentally.
Well, so he's saying that you have a neutral model of development.
No, I'm not saying that he has, but this would be one way of having, without implementing GRNs, if we increase that, the likelihood of having any change in development is equal in any direction. So that you have no preferred tendency to change in development. So the phenotype could go anywhere.
Yes. But I agree, I see your point. I have to understand this better. I would like to indeed to like implement explicitly a set of paths. You have several paths to move. And instead of a quantitative value, you have and each path contains a probability given a genetic architecture, something like that.
This has to do with something we talked last November, that to implement the contrary, not developmental drift, but developmental preferred changes. You could do this at the phenotypic level by association frequencies between different phenotypes. If you make the phenotype be clusters, like if this changes, is likely to change too. If this changes inversely, we've all implemented a genetic regulatory network.
By this addition in two individuals?
No, two traits in the same individual.
And the same individual, two
correlation between traits in the same individual. So if you do this, you go against drift. Then you can implement some developmental bias without implementing a genetic regulatory network. I'm just worried because this is already complicated enough to put one layer of genetic regulatory network.
Yeah, but maybe you could have some traits and not only one.
No, you have to have some traits to
Yeah.
do this, I would say. Because if you have some traits, then you can, what would be developmental drift? The frequency of changing these traits completely independent from all other traits and the same for all traits. And you would move in a way from developmental drift when you get association frequency. This changes in one direction or the other. So we could simulate this way. But
So
you have to have more traits.
What is
But you
not
have to have more traits.
What is not difficult to do is that you have several traits. You can put linkage,
Yeah, exactly.
and you can have that fluctuations, right, are correlated with the linkage strength.
That's
This-
much easier than doing a GRN, a genetic
Yes.
regulatory network.
Yes. Maybe you had a question already.
Right now you have multiple loci, but they are coding for only one trait.
Yes. This is what we have now.
It's quantitative. It's polygenes.
Yes, polygenes, yes. But just determining one trait.
More questions? Okay. So thank you, Carlos.
Thank you.